Good morning. As president of Pi Mu Epsilon, the National Math Honor Society, I am delighted that Dr. Daniela Witten will be our 49th J. Sutherland Frame Lecturer. It was actually Dr. Frame himself who gave the very first Frame lecture at the AMS MAA summer meetings in 1975, 50 years ago. She's the 49th lecturer. We missed one year because of COVID, so it almost lines up perfectly year for year. For those who don't know, Dr. Frame served as president of Pi Mu Epsilon from 1957 to 1966. He was a prolific researcher, and he was dedicated to helping students grow professionally and achieve success in their careers. It's therefore fitting that Dr. Witten is this year's Frame lecturer, as her own pursuits are very much aligned with Dr. Frame's. Dr. Witten received her PhD in statistics from Stanford University and currently serves as professor of statistics and biostatistics at University of Washington, Seattle, and as the Dorothy Guilford Endowed Chair in Mathematical Statistics. She's a prolific researcher where she works to develop statistical machine learning methods for high-dimensional data with a focus on unsupervised learning. She's received numerous awards and honors for her work, including the following. She's a fellow of the ASA and the Institute for Mathematical Statistics and an elected member of the International Statistical Institute. She's a recipient of the NIH Director's Early Independence Award, a Sloan Research Fellowship, an NSF Career Award, and a Simons Investigator Award in Mathematical Modeling of Living Systems. She received the President's Award from the Committee of Presidents of Statistical Societies, awarded annually to a statistician under the age of 41 in recognition of outstanding contributions to the field of statistics. She also received the Spiegelman Award from the American Public Health Association for a statistician under the age of 40, who has made outstanding contributions to statistics for public health, and the Leo Breiman Award for contributions to the field of statistical machine learning. In addition to being a co-author of the very popular textbook, Introduction to Statistical Learning, she serves in an editorial capacity for several journals, including Biometrica, the Journal of Computational Graphical Statistics, the Journal of the American Statistical Association, the Journal of Machine Learning Research, and the Journal of the Royal Statistical Society Series B. And Pi Mu Epsilon would like to add one more honor to this list, if you're willing. We'd like to induct you officially into PME, so please repeat the pledge after me. I solemnly promise.
I solemnly promise
that I will exert my best efforts
to
promote true scholarship, particularly in mathematics and statistics, and that I will support the objectives of the Pi Mu Epsilon Honor Society. Congratulations.
Thank you so much.
And please join me again in welcoming Dr. Witten as she tells us about selective inference for real-world problems.
Thank you. Well, thank you so much. It's such an honor, and for those who can't see the small font, I'm specifically a member of the Alabama Gamma chapter, which just feels like extra special, so this is such an honor. Okay, so I'm excited to tell all of you today about my work on selective inference for real-world problems. And this is a line of work I've become very interested in the last few years, and my hope for today is to explain to you what I mean by selective inference, why this is something that we need, and sort of two different avenues that we can take to try to address some of these problems in practice. Okay, so I'm a statistician, and all the stat textbooks are kind of written the same about an idealized reality where there's a researcher who asks some question, which as a statistician will frame that in terms of a null hypothesis that I'll call H naught. And then the researcher, after asking that question, collects some data, analyzes the data to either reject or else not reject that null hypothesis. If the researcher fails to reject the null hypothesis, then she's very sad and she goes home maybe. But if she rejects it, then it's really great. Maybe she has a party and she publishes her results. So this is what we write textbooks about. And this paradigm is the setting in which all of classical statistical inference operates, where specifically the null hypothesis, the scientific question, is specified in advance before the data is ideally collected. Ideally before the data is even collected. By contrast, the real world often is a lot messier. Somebody might collect data, then based on the data, she might take a little peek, ask a question, formulate a null hypothesis, either reject or not reject that null hypothesis. If she's not happy with her results, she might iterate with a different question, different null hypothesis. And then finally, publish results. So this is a problem, but this is often how science is done. Here's a different way that I think about it, which is like we write textbooks in this fictional reality where before we see our data, we decide exactly what hypotheses to test and we stick to the plan. And we don't live in the fictional world, we live in a real world. And the fact is that what we often do is we decide what hypotheses to test after I look at my data. And I just want to be clear, like, I'm not criticizing people for living in the real world. Rather, what I'm saying we should do is make statistics applicable in the real world in which we are all actually living. So why is it that the real world is really different from how the textbooks are written? Well, I think that specifically in recent decades, there's been a big shift towards hypothesis generation as our ability to collect data and store it and analyze it has really increased. So as data sets continue to grow in size, it's just natural that instead of coming up with one pre-specified hypothesis and collecting data just to answer that one question, it's natural that if we're going to have really large data sets, we would start to just like, if we can collect the data, why not? Let's just collect the data, see what's there. So this naturally leads to a shift towards data-generated hypotheses. It's natural that if we collect all this data, we might find something interesting. We might notice some kind of trend, and then we'd want to use that same data to confirm that it really is interesting, that we didn't just notice something that's there by chance. So in other words, what's happening today is that we're more and more generating hypotheses based on the data and then testing the hypothesis on the same data. And I'm going to just refer to this as double dipping, this very real practice where you dip into your data once to generate a hypothesis and then again to test that hypothesis. So here's an example of a paper from neuroscience. This is a review article published in Nature Neuroscience, actually from 2009, where they talk about circular analysis in systems neuroscience, the dangers of double dipping. And you can see that they define double dipping as the use of the same data set for selection and selective analysis. So that's exactly what I'm referring to here. This practice of using the data both to select a hypothesis or decide what question we want to ask, and then try to answer the question on the same data. And here's an example from Genomics. This is a review article that was published a few years ago in Genome Biology. They describe 11 grand challenges of single-cell data science. And just bird's eye view, single-cell data science, it's like single-cell data is one of like the big types of data to have come out of biology in the last decade, and it'll relate to the application that I'll show later in this talk. They describe 11 grand challenges in the entire field, and one of the challenges is in fact exactly double dipping. In fact, it's the exact application that I'll get to later in this talk. So this is a real problem. This isn't an instance of like statisticians just like standing on a hilltop telling scientists that they're doing things wrong. This is a situation where statisticians see a problem and also the scientists see a problem. These are articles published in Nature Neuroscience and Genome Biology by the actual scientists on the ground who know that there's a problem with how these analyses are being done because they know that they're analyzing the data, or rather they know that they're using their data twice, both for hypothesis generation and for hypothesis testing. So the scientists know that there's a problem, the statisticians know that there's a problem. The question is, can we as statisticians pose a solution that allows scientists to conduct valid statistics based on this very natural data analysis pipeline for the world in which we live? And so that's the focus of my talk today. So just to be completely clear, what's wrong with double dipping? Why am I not allowed to generate a hypothesis from the data and then test that hypothesis on the same data? Well, just very straightforwardly, classical statistics, like all those things that you learn in your intro stats classes, require that the null hypothesis be specified in advance before looking at the data. And in particular, if you double dip, not only is your null hypothesis not specified in advance, but your null hypothesis itself is actually a function of the data. So your null hypothesis is random. And that's just that just breaks classical statistics. That's just not allowed, is really the simplest way to put it. And in particular, as we're going to see, the way in which it breaks is that even when the null hypothesis holds, we're going to end up with p-values that are way too small. And I'll talk about that in a minute. So today I'm going to talk not about two strategies to avoid double dipping, but actually two strategies for valid inference after double dipping. Because I think that the idea that we need to use our data to generate hypotheses and then test those hypotheses, that's not going anywhere. So rather, let's find ways to do this that are valid. I'm going to talk about two different strategies. The first one, conditional selective inference, and the second one will be data thinning. And these are really two quite different approaches to get at the same goal, which is enable a researcher to generate a hypothesis and test that hypothesis based on the same data. Okay, so first I'll talk about, of course, strategy number one, conditional selective inference. I have this little recycling symbol here. And the reason that I have a recycling symbol is because the point of the strategy is actually we are going to use the data twice. We're literally going to use the same data to generate a null hypothesis and test that null hypothesis, but we're going to do it in a very particular way that accounts for the fact that we're reusing the data so that we're actually going to be able to conduct valid statistical inference. Okay, quick reminder, what is a p-value, just so we can all be on the same page. A p-value is the probability of seeing a result as extreme as the one that we saw in our data under the null hypothesis. The thing that makes it a p-value is that if the null hypothesis holds, it needs to follow a uniform 0-1 distribution. So if you tell me that the null hypothesis holds and you got a p-value of 10 to the negative 7, like, I'm not going to say that's impossible, but I'll say the probability of that was 10 to the negative 7, if that makes sense. You know, probably if the null hypothesis holds, your p-value should be like uniform 0-1. So I'll be a little skeptical that things are working out for you. And this is really important because if your p-value is not uniform 0-1 under null hypothesis, then bad news, it's not a p-value. Like, this is the actual definition. This is what makes it a p-value. And this is also referred to as type 1 error control. So if you hear me say that, what I'm saying is that under the null, I need my p-values to be uniform 0-1. So this is a need to have. This is just what makes it a p-value. If this doesn't hold, you can call it a p-value if you want to, but you shouldn't because it's not one. And then we also have a nice to have. The nice to have is that if the null hypothesis doesn't hold, then the p-value is small. The reason that this is nice to have is because the way we use p-values in practice is that if we get a small number like 10 to the negative 7, then we're going to reject the null hypothesis. So this wouldn't be a useful procedure if the p-value weren't small under the alternative. But this is a nice to have in the sense that even if this doesn't hold, you still have a p-value, it's just not useful. Okay, so the main point of the slide is what we need is for our p-values to be uniform 0-1 under the null, and hopefully small under the alternative, but we really need them to be uniform 0-1 under the null. To be clear, if I just like use a random number generator in R just to like generate a uniform random variable from 0 to 1, and it's less than, like randomly generated number is less than 0.05, and that causes me to reject, like that was a valid p-value because it's just uniform 0-1. It's just not useful. So we'd also like it to be useful, but it's got to be uniform 0-1 under the null. Great. Oh, and I just want to say one other thing actually still on this slide, which is if you're not into p-values, if you think p-values are not the way to assess statistical significance, if you believe in other measures of quantifying effects like confidence intervals or whatever it is, like God bless, all the ideas in this talk remain. For example, there's a direct duality between p-values and confidence intervals. I'm just going to talk about p-values here because it's like very easy to check if it's a valid p-value. But if your p-value is valid, then your confidence interval is valid. There's a one-to-one mapping. So we're just going to use p-value as a placeholder for your preferred way to conduct statistical inference. Okay. And in this talk, I'm going to be showing an example that has to do with hypothesis testing after clustering, or actually also hypothesis testing after principal components analysis. That'll be kind of a motivation for where double dipping comes up in practice and how we can solve it. Okay, so hypothesis testing after clustering, what am I talking about? Here's some simulated data. I just simulated 100 observations independently from a normal 0-1 distribution on the x and y axes. And then I clustered the data. There's no true clusters in this data, right? It's just, there's all like normal 0 identity. But I can cluster the data, just like an algorithmic operation. I can cluster anything. And if I cluster this data, here's what I get. These are my 3 clusters. And then what I'm going to do is I'm going to say, hey, I wonder if there's a difference in the population mean between the green and the orange clusters. And I'm just going to compute a p-value for the difference in means without acknowledging the fact that the hypothesis of a difference in means between the blue and the green clusters was generated from the data. I'm just going to blindly compute a p-value as though the hypotheses were specified in advance. So I just take out my ruler, I compute the distances between each pair of clusters, divide by the standard error, and all 3 of my p-values are less than 10 to the negative 7. So I have this really unhappy, scared-looking face, and the reason I'm unhappy and scared is because the null hypothesis holds here. There's no true clusters. So clearly there's a problem with this procedure. And of course, we now know the problem with the procedure is that we double dipped. We generated the null hypothesis here after looking at the data. That's not allowed. So how can we fix this? Oh, and again, if you don't like p-values, it's fine. Just every time I say p-value, just use the word confidence interval in your head. Okay, so was that just like an unlucky simulation, or was this something systematic that's going on? And the answer, of course, is that this is very systematic. This type of plot is called a QQ plot, and it shows the distribution of p-values over 10,000 simulated data sets, all simulated in the same way with the same procedure. And essentially, it's comparing the distribution of the p-values that you get to the uniform 0, 1. Remember, my p-values should follow uniform 0, 1. These ones don't, because those quantiles are not on the diagonal. The p-values are not uniform. They're way too small. They're like 10 to the negative 7 instead of 0.5 or whatever. And the way that I'm going to refer to this is that the type 1 error rate is 97%. So if I decide that I'm going to reject the null hypothesis of no true clusters, anytime I get a p-value less than 0.05, then I'm going to actually reject 97% of the time instead of 5% of the time. That's really bad. And you might notice I put the word selective here in front of the word type 1 error rate. I'll go into more details about that in a couple minutes, but the idea is the word selective is to indicate that this is the type 1 error rate about a hypothesis that was selected on the data. Okay, so this is the issue. And now we're going to show how we can use conditional selective inference to solve this problem, in other words, to get valid p-values. And again, conditional selective inference, that's the first strategy I'll be talking about today, where I had like the recycling symbol. So the idea is we're going to actually use our data twice. We will use our data both to generate a hypothesis and to test it, but we're going to account for the fact that we've used the data twice in order to conduct valid inference. So how can we do this? Just to be clear, we're trying to answer the question, are the cluster means really different in the population, where I've clustered the data in order to get those cluster means, or in order to identify the clusters. And to make this more statistical, suppose that I have an n by q data set x, of which each row is independently drawn from a q-dimensional normal distribution, where here q is the number of features in my data, so like the number of variables that I've measured. Each observation has its own mean vector, mu sub I. And just for simplicity of exposition in this talk, I'm assuming that the covariance for each of the observations is a multiple of the identity. But like, it's fine if it's not. It just, we would need to have a bunch of sigma inverses floating around. Okay, so what we're going to do is we're going to cluster the rows of the data set in order to get C1 hat through CK hat. So like C1 hat contains the indices of the observations assigned to the first cluster. And C1 hat through CK hat just partition the indices 1 through n. And then we're going to take mu bar CK hat to be the mean, the population mean, so the mean of the mu i's, or rather the sample mean of the mu i's for the observations in the kth estimated cluster. So remember, mu I is a population quantity. Mu I is the true population mean of the ith observation. And mu bar CK hat is sort of like the sample mean of those population means. So mu bar CK hat is a funny quantity. And the reason that it's funny is because it looks like a population quantity because it has a mu in it, but it also involves the data because it has a CK hat. And remember, CK hat was the clustering we got from the data. So right away, like our spidey senses should be telling us that there's a problem, and indeed this is exactly the problem, is that the parameter we're interested in is not a well-behaved statistical parameter since it involves the data. But we're just gonna try to go for it. We're gonna try to accomplish this goal, which is testing this null hypothesis, and the first thing we're gonna have to do is a little bit of work to make this null hypothesis meaningful statistically, because the entire problem with double dipping is that actually, as written here, it's not a meaningful null hypothesis because it involves the data. Okay, so the first thing we're going to do is like what I'm going to call in this talk like a naive approach to get a naive p-value. And what that would be is like the scientist who just like pretends that they didn't come up with the clusters on the same data and just like runs a t-test as though that were an okay thing to do. And the way that you would do that is to test this null hypothesis. You would just kind of ignore the fact that CK hat and CK K prime are functions of the data, and you would just calculate the probability of observing such a big difference between the means of the observations in the kth and k prime clusters by chance under that normal distribution assumption that we had. And we can compute that probability. This is sort of just like standard statistical inference, using the fact that each observation is IID, or rather independent normal mu I variance sigma squared. This is just the probability that chi-squared random variable with q degrees of freedom exceeds some threshold. I know how to calculate that using just like the quantiles of the chi-squared distribution. And as we saw already, if you do this, this is not going to give you a p-value that's uniformly distributed under the null. In other words, this is not a p-value. And this is exactly what I showed you in that QQ plot. Like when I told you the p-values were 10 to the negative 7, this is what I did. So we should not do this. OK, so what should we do instead? Well, what we should do instead, it turns out, is we can fix the problem by conditioning on the hypothesis selection procedure. Or put another way, let's account for the fact that the data was such that we became interested in this particular hypothesis in the first place. So we're going to define the selective p-value to be the probability under the null hypothesis of seeing such a big difference between the observation in the k and k prime clusters, given that clustering the data gave me c hat k and c hat k prime. And why would I want to condition on that? Well, I think one way to think about the intuition is that the only reason you're interested in this particular null hypothesis is because clustering your data gave you these two clusters. If clustering your data hadn't given you these two clusters, you would have been interested in some different null hypothesis. So we should account for the fact that the data made us interested in this null hypothesis in the first place. And that's what that conditioning event does. So just to say it in plain English, we're asking the question, out of all data sets that result in these two particular clusters, what's the probability, assuming that there's no true difference in their means, of seeing such a large difference in the sample? And the thing that makes this a selective p-value rather than a regular p-value is the text in blue. Like the naive p-value, just you'd cross out the text in blue. OK. So what are the properties of the selective p-value? Well, it controls the selective type 1 error rate, which is the probability of rejecting the null hypothesis if the null hypothesis holds, given that we decided to test this null hypothesis. So usually a p-value tells me out of the universe of all particular data, usually a p-value says out of the universe of data sets that could arise under this null hypothesis, what's the probability of a false rejection of the null? And here instead I'm saying, instead of looking at the whole universe of data sets that could arise under this null hypothesis, I'm going to subset the universe to the data sets that would have given me this null hypothesis. And this controls the selective type 1 error. In other words, it gives me a valid p-value that accounts for double dipping. So it sounds like I should be done. I'm not. The reason I'm not done is because like I'm not a mathematician. I'm a statistician. That was a joke. Sorry. Wrong crowd. I need to be able to compute it. And this is just like, I can write this down, but I need to be able to do this computationally. And so that actually turns into the big challenge. So even though this is a valid selective p-value and I can prove a theorem saying that it's valid, it's only useful to me as a statistician if it's something that I know how to calculate on a data set. And it turns out that actually that selective p-value is not something that I know how to calculate on a data set. For various technical reasons that I'll get into, it's not something that can be calculated. So in order to get something that I can actually calculate, I need a condition on a bit more information. And I'm not going to write down what I'm going to calculate more. I'll just instead show you what the effect of it is. But it's just like some linear algebra, some projections. And I'm going to condition on a bit more. And I'll tell you that like it's not hard to prove that if we condition on more, we still have selective type 1 error control. So we're still going to get a valid selective p-value. The reason we're going to condition on more is in order to make this something that's like mathematically and computationally tractable. And once we condition on more, our p-value is going to take a slightly different form, which is it's going to be the probability that a random variable phi exceeds some threshold, given that clustering x prime of phi gives me the clusters that I observed in the data. So there's two new things that popped in here. One is phi, one is x prime of phi. I'll just tell you in words that phi is a random variable that follows a chi distribution with q degrees of freedom. And now I'm going to explain to you what x prime of phi is. It turns out that x prime of phi is going to be just like our original data, but with a shift. So I'll show you what that looks like. So here's the definition of x prime of phi. But if it's easier to see it in a picture, if this is our original data, suppose we have some data, got 3 clusters on it. I'm interested in the blue versus the orange cluster. I'm just going to look at the centroids for those 2 clusters. And if phi is big, then I'm going to just generate x prime phi by pulling those 2 centroids apart. And if phi is small, I'm going to get x prime of phi by pushing those 2 clusters together. And if phi is equal to the observed distance between the 2 clusters on the real data, then x prime of phi is just equal to x. So again, x prime of phi is just my original data but where I've pulled apart or pushed together the two clusters of interest. So to summarize, I have a selective p-value, which is just the probability that a chi-distributed random variable exceeds some threshold, given that clustering the data gives me the clusters of interest, where I don't need to think about the whole universe of data sets. I don't even need to think about the universe of data sets that would have given me C at k and C at k prime. I'm just going to think about the universe of data sets that look like the original data, but have either pulled or pushed the two clusters of interest. And what this is telling me is that out of all of the data sets that look like the original data, but the two clusters of interest have either been pulled apart or pushed together, out of all such data sets that give me the clusters of interest, what's the probability under the null hypothesis of seeing such a large difference between those clusters? And this is a valid p-value. We can prove that it maintains type 1 error control. So we're almost there. We just need a way to calculate this. And now we went from before having a selective p-value that just like mathematically was not going to be something that we could feasibly think about, to now a p-value that, like, in principle, we should just be able to compute this p-value. Like, for example, if I've told you that phi is a chi-distributed random variable with q degrees of freedom, like the square root of a chi-squared random variable. So in principle, if I wanted to approximate this p-value on a computer, I could just like draw phi from a chi-squared, or rather a chi-distribution, check whether x prime of phi when I cluster it gives me C at k and C at k prime, and do that a million times, and then I would get like a great approximation to this p-value. So this is something that 100%, if I have like enough computation resources, I could approximate with Monte Carlo. But it turns out actually that for a lot of types of clustering that are of interest, like some of the really common algorithms like k-means clustering and hierarchical clustering, we've actually proven that you don't need to use Monte Carlo. We have sort of an analytical solution to this, where you can, like, in actually no more computation than it takes you to cluster the data in the first place, we can analytically compute this p-value. But if you didn't want to have to do it analytically, it is something you could do with Monte Carlo if you had enough compute power. Okay, so now I'm going to show you what sort of results we get if we do this. So again, we're going to compare on a data set the naive p-value that we know is not valid, that arises from double dipping, to this corrected selective p-value, which provably has the right properties that make it a valid p-value. And we're going to look at this data set of polymer penguins. So we have 3 types of penguins: chinstrap, gentoo, and Adelie penguins. And we're going to only look at female penguins, and we're going to just look at the ones for whom we have data on their species, their bill length, and their flipper length. So we have bill length on the x-axis, flipper length on the y-axis, and the shape here tells me what type of penguin it is. So I happen to know that there's 3 species of penguins, but I'm going to run an analysis without using this information. And in particular, I clustered the data using sort of a standard clustering algorithm. I find 6 clusters, which are the 6 colors here. And now I'm going to I'm going to consider the null hypothesis that says that there's no difference in the means of these 2 clusters. In other words, like, the difference that I'm seeing here is just what you'd expect by chance, given that those clusters were estimated from the data. There's no population difference. Now remember, I happen to know the species of these penguins. So I happen to know that these are all Adelie penguins. So that makes me think that probably the difference that I'm seeing here is actually just due to chance. So what I would like to see is that my selective p-value is not particularly small here, because I think that this difference between these Adelie penguins is just chance. And indeed, if I run this analysis, the naive p-value that doesn't account for the fact that I double dipped is quite small, smaller than 0.01. It would cause me to reject the null hypothesis. Whereas my selective p-value is 0.87. It's pretty much saying, hey, like, that's just chance. Okay, so that's reassuring. Now we can say, what about these 2 clusters? Remember, I happen to know that the circles are Adelie penguins and the squares are gentoo penguins. So that makes me think that, like, probably there is a real difference between the flipper and the bill lengths here. But I'm not telling that to my analysis procedure. I'm just going to see what the data tells me. And if I do that, then the naive p-value is tiny. It's 10 to the negative 16. But also the selective p-value is tiny at 10 to the negative 15. And so this is really nice, because what this is saying is that, like, I don't care as a statistician, 10 to the negative 16, 10 to the negative 15. Either way, I'm rejecting the null hypothesis. So the point is, in a setting where there really is a difference between the means, in other words, when the null hypothesis does not hold, my selective p-values are successfully rejecting the null hypothesis. In other words, I have power. So I have valid p-values that account for the fact that I've used the data twice, that are powerful under the alternative, and that are valid under the null. Great. So now I just want to go on to the other strategy that I'm going to talk about in this talk. So that was conditional selective inference. Remember, conditional selective inference, I have this like recycling symbol because we're actually going to use the data twice, but we're going to account for the fact that we've used it twice, right? And by contrast, data thinning, we're not going to use the data twice. We're going to actually split up the data. So I have you can either put it in the compost or the trash or the recycling. You've got to choose one. So that's the analogy I'll be using for data thinning. It's going to give us a way to split up our data so that we actually don't use it twice. So what does that look like? What I'm going to do is I'm going to split the data into independent training and test sets. Then, okay, that slide got cut off. Then I'm going to select the null hypothesis on the training set, and you'll never guess what comes next. I'm going to test it on the test set. This is what happens if you make last-minute edits to your slides while you're sitting right there before your lecture begins. Okay. All right, so the best-known approach to obtaining independent training and test sets is called sample splitting. And I'm sure many of you are familiar with sample splitting already, but the idea is suppose I have 5 observations. I'm just going to split them into 2 sets. I'll call one of them a training set. I can use it to select my null hypothesis. Like I can just like knock myself out on that training set, do whatever I want to it. And then I'm going to test that null hypothesis on the test set, and I'm good to go, as long as the training and test sets are independent. I don't need to worry about double dipping. And one way to think about the fact that I don't need to worry about it is that, yeah, the null hypothesis was selected based on the training set, but the training and test sets are independent. So the null hypothesis, like I can think about testing the null hypothesis on the test set as independent of the data used for training, and I'm good. I don't need to worry about the randomness in that null hypothesis, if that makes sense. So sample splitting is great. If you're familiar like with cross-validation, then this forms the basis for cross-validation. Sample splitting is just like truly the #1 tool in the toolbox of data analysts in a lot of settings. It's great except when it's not. Sometimes sample splitting fails pretty spectacularly. So one example is if your sample size n is small. So like in that example, n is 5, you wouldn't actually want to do sample splitting. I mean, okay, I hope your n is bigger than 5, because it's hard to do a lot of statistics if n equals 5. But I mean, there are settings where you can. But regardless, if you had 5 observations, I think you'd be pretty bummed if you had to divide your data into 2. It's not great if the observations aren't identically distributed, and I'm not going to get into details today, but essentially like an inherent assumption to sample splitting is that all your observations are drawn from the same distribution, and if they're not, it's not quite exactly clear statistically what you've accomplished with sample splitting. It's a little bit problematic. Most importantly, though, sometimes it just fails spectacularly. And the time that it fails spectacularly is if your observations are not independent. So like here, the whole reason that sample splitting would work is if my 5 observations are independent, then I get a training set and a test set that are independent of each other. But if my 5 observations aren't independent, then I don't get an independent training and test set, and I haven't solved my problem. That's actually not the only setting where sample splitting might not work. Another example of where sample splitting will not work actually is that clustering example that we saw earlier. So I'm going to come back to that later. So today what I'm going to talk about now in my strategy #2, which I'm calling data thinning, is a new way to split our data into independent training and test sets that will work even if our sample size is small, even if our sample size is 1. It'll work if the observations are not identically distributed. And in particular, we don't need to worry about the observations being independent. So it's going to solve these problems in a range of settings. Okay, so what is it that I'm going to try to accomplish? Well, again, I'm going to start with, let's say, 5 observations. But I actually want a picture that looks like this, where I now have x superscript 1 and x superscript 2, where x superscript 1 is going to be my training set, x superscript 2 is going to be my test set. And I'm going to want x superscript 1 and x superscript 2 to be independent. And to frame this a little bit more statistically, suppose that I have a random variable x drawn from some distribution piece of theta where theta is unknown. I'm a statistician, so there has to be an unknown parameter, and it's theta. So for the rest of this talk, anytime you see theta, it's an unknown parameter. And my goal is to get an x1 and x2 such that x is equal to t of x1, x2, where t is a function, a deterministic, so I mean non-random function, that allows me to get x back from x1 and x2. And furthermore, x1 and x2 are independent. So if I could do this, then I would be great, because I'd have an unknown parameter, theta, and even without knowing theta, I'd have a way to take x and split it into independent x1 and x2. So that is my goal. Just because I can write down a goal doesn't mean I can accomplish it, so we'll have to see if this goal is feasible. But again, to emphasize, the distribution of x involves some unknown parameters that I'm calling theta. So here's just like a ray of hope that this might be something that we can accomplish. So suppose that x comes from a Poisson distribution with some unknown mean theta. So then I'm going to define x1, or rather I'm going to get x1 by drawing from a binomial distribution with mean x, rather size x and probability epsilon, where epsilon is just a user-specified number between 0 and 1. And then I'll just take x2 to be x minus x1. So notice this is something that I can do. This is a procedure that I can carry out even without knowledge of theta, because there wasn't any theta required in the expressions for x1 and x2 here. And it turns out that if I do this, then first of all, by construction, x is equal to x1 plus x2. But more interestingly, x1 and x2 are independent. And it turns out also that x1 and x2 themselves are each Poisson with distributions that involve that unknown theta scaled by epsilon and 1 minus epsilon, where epsilon was the user-specified parameter. In this talk, if you want, you can just set epsilon equals 1/2, because it's a number that you get to choose. So this is really nice, because we exactly accomplished our goal of taking x and splitting it into x1 and x2 without knowing theta, where x1 and x2 are independent, and also have distributions that depend on theta. So for example, instead of generating a hypothesis on x and testing that hypothesis again on x, I could generate a hypothesis on x1 and test it on x2. And because x1 and x2 are independent, I would avoid all those issues with double dipping. OK, so this is cool, but there's a couple of problems. Number one is most data is not Poisson distributed. So this seems very limited in scope. And also another thing that I need to clarify is this is not a new result. This is binomial thinning of a Poisson random variable, like you see it in like a senior level undergrad statistical inference course for like a stat major, or else like in a first year grad class. This is not new. This is probably 100 years old, although I actually don't know the original citation. But not only is it not new, in the last few years it's actually been used in the context of selective inference. So even the application of this old result to selective inference is not new. But it's a good proof of concept that our goal might be achievable. So now, the Poisson distribution is great. Why don't we think about another distribution that's also pretty easy to work with, which is the Gaussian distribution. So now suppose that x is Gaussian, and remember I said in this talk theta is unknown. So the mean here, theta, is unknown. The variance is actually going to be known here in what I'm about to show you. So we have x from a normal distribution with unknown mean. I'm going to draw x from a normal distribution with mean epsilon x, variance epsilon 1 minus epsilon sigma squared, where again, epsilon is a number you can choose. You can just set it to be 1/2 if you'd like. And then I'll just define x2 to be x minus x1. And if I do this, of course, by construction, x is equal to x1 plus x2. Furthermore, x1 and x2 are independent. And also, just like a fun fact, x1 and x2 themselves also follow normal distributions that look a lot like the distribution of that original x. And I was able to do this kind of magic trick without knowing theta, which again is a key point, because if I had to know all the parameters, there wouldn't be any need for statistics, right? OK, so this again is very interesting that it can be done. It generalizes this very old result about Poisson random variables to Gaussian random variables. This has been used in the selective inference literature since at least 2018. But it does give us a little bit of hope that maybe there's some general principle here that extends beyond just Poissons and Gaussians, that might be something we could apply sort of in settings where our data is not necessarily Poisson or Gaussian. OK, so what we want to know is, are those somehow special cases, or is there a bigger principle at play here? And we're going to try to generalize this procedure using just a little recipe, which is we're going to imagine that we have x1 and x2 that are independent. x1 is drawn from some distribution q sub theta 1. x2 is drawn from distribution q sub theta 2. I should mention that if you're not a statistician and you're in the audience, this is like the most statistical slide that we're going to have, I think. We're going to take t of x1, x2 to be a sufficient statistic for theta, where if you don't remember, if you haven't seen sufficient statistics, it's just like a function of the data that summarizes all the information about the unknown parameter, so that once you condition on the sufficient statistic, your data no longer has any information about that unknown parameter. So I'm going to define G sub t to be the conditional distribution of x1, x2, given that sufficient statistic. And just by definition of what a sufficient statistic is, this conditional distribution does not depend on theta. That's just the definition of sufficiency. And then I'm going to let p sub theta denote the distribution of that sufficient statistic. And what that means is that I can thin the family of distributions p, indexed by the unknown parameter theta, by the function t, or to put it in plain English, if you give me a random variable x distributed p sub theta and you don't know theta, then I have a strategy that I can carry out in practice that will allow me to sample x1 and x2 from the conditional distribution g sub x. And that'll give me x1 and x2 that are independent and that each follow distribution q theta 1 and q theta 2. So this is actually exactly the procedure. If you plug in like the Gaussian and the Poisson examples, this procedure gives you back those two examples. But it turns out this is actually a very general procedure. So this is a table from one of our papers where the first two rows are those two examples that I've already shown, which were known in the literature. And the rest of the table is new. And so what's going on here? Well, on the top, we have distributions in the natural exponential family. These are kind of like the best behaved statistical distributions that are the nicest. And for example, if you give me a binomial random variable or a gamma random variable, then I can thin it into k, not just two, but actually k independent components that each follow a gamma distribution. And the way that the last column of the table is telling me how I'm going to recombine those independent pieces to get back the original data. The answer is I'll use addition. Now, if we move down a little bit, for example, into the general exponential family, which is a slight generalization of the natural exponential family, we could look, for example, at like the Pareto distribution with parameters nu and theta, where theta is unknown. It turns out it's pretty goofy, but if you give me a Pareto random variable with parameters nu and theta, where theta is unknown, I can give you back capital K independent gamma random variables with that same unknown parameter theta. And if you want to know how to recombine those gamma random variables to get back the Pareto, this is goofy, but you take nu and you multiply by the exponent of the sum of those gammas. So the details are a little bit finicky depending on the distribution, but the point is this table really encompasses like a whole bunch of different things here in terms of the natural exponential family, the non-natural exponential family, distributions outside of the exponential family like the uniform. We can even think about applying this strategy to an arbitrary non-parametric unknown distribution f, for which we have NIID observations, and sort of magically, if you apply that recipe, you end up back at sample splitting. So this allows us to view this new data thinning framework as a generalization of sample splitting, where that generalization, or rather where that special case of sample splitting is what arises if your observations are independent, identically distributed, which we know. We know that sample splitting only makes sense if you have IID observations, and indeed that's what we find. Okay, great. So another really neat thing about this thinning framework is that there's a very natural decomposition of the Fisher information. So Fisher information is the statistical term for like the information that the data has about an unknown parameter. And remember, I told you there's this user-specified parameter epsilon. For example, you can take it to be 0.5. If you do that, then 50% of your information will be in your training set, 50% will be in your test set. If you set it to be 0.8, you'll do an 80-20 split. So it's just really nice that when you do thinning, you know exactly how the information is going to be allocated. And that's actually different from sample splitting for reasons that I'm not going to go into, but essentially thinning can give you higher power than sample splitting because it cleanly allocates the Fisher information in a way that sample splitting does not. And then finally, there's a lot of distributions we can thin, including quite a few that aren't shown in this table. There's one distribution that you cannot thin, which is a Bernoulli. So what this is saying is that if you have just one Bernoulli random variable, remember Bernoulli is either 0 or 1, you cannot thin it into two independent pieces. And that's kind of intuitive, because like, if your x1 equals 1, then your x2 better equals 0, because they got to sum, you know what I mean? They got to recombine somehow to give you either a 1 or a 0. There is an actual real proof that goes beyond the words I just said. But it is sort of intuitive that like with just one bit of information, how can you get two independent pieces of information that are non-trivial? You can't. So if you give me 10 Bernoullis, I can thin them, but I can't thin just one. Okay, so remember our goal was a way to split our data into training and test sets that are independent. Data thinning gives us a way to do that, and now I'm going to show how this is useful in practice so that we can select the null hypothesis on the training set and test it on the test set. So the last thing I'm going to talk about just for the last few minutes is an application to data thinning. And what's fun is like sometimes as a statistician, you like do all the math in your paper, and then you're like, you wrote up the whole thing, and then you're like, shoot, I need like a data set to show how this works. And that's not what happened here. What actually happened here, the way that I got interested in this whole line of work on data thinning, I had already done the work on selective inference, and that was a line of work for my group for a few years. But then during COVID, I was like taking advantage of the fact that all the talks were on Zoom. So I was giving a seminar to some department that I didn't have to fly to the East Coast for, so it was very convenient. I could like be in my home office in Seattle on Zoom, giving a talk to some computational biologists, and one of them like drew my attention to this particular problem, and actually all this line of work on data thinning came out of this application. So single-cell RNA sequencing data, I mentioned it at the beginning of this talk, is like a very big new type of data in biology in the last 10 years that allows you to measure the activity of many, many, many thousands of cells, all of their genes. So, you know, tens of thousands of cells, 20,000 genes, lots of data. And it's so much data that there needs to be some kind of like way to simplify it a little bit. And so there's sort of a simple two-step paradigm that scientists usually use when they're thinking about this data, which I'm going to show a cartoon of. So step one, what they do as scientists is that they identify some kind of latent structure in the data, perhaps with clustering or perhaps with what they call trajectory inference. But I'm a statistician. I'm going to call this PCA. You can think about it as a generalization of PCA. But what they do is they find some kind of latent structure in the data, and then they perform what they call a differential expression analysis, where they test each gene for association with that latent structure. So for example, in the case of clustering on the left, they might test for a difference in the means of clusters 11 and 17. That's just like the example we saw in the first half of this talk. Or on the right, if they did PCA, they might test each gene for association with the first principal component. In either case, if you just do step 2 using like a standard t-test or something, you're in trouble for exactly the reasons we've been talking about for the last 45 minutes. So here's a paper published in Nature. They released a Shiny app that allows you to perform this two-step analysis. Here is an example of what comes out of the Shiny app. They cluster the data. Remember, this data is like 20,000 dimensional because there's 20,000 genes. This is a two-dimensional representation of that 20,000 dimensional data, which is why these clusters look a little goofy, but because we're looking at just a two-dimensional representation of high-dimensional data. But they cluster the data. They come up with these clusters. And then there's a little drop-down menu. Choose cluster 11, choose cluster 14. And what it gives you is a bunch of p-values. It actually gives you 20,000 p-values like this. Each one is for the difference in means between the 11th and 14th cluster. So the H0J is the expected, is the null hypothesis. The expected expression for the J gene in cluster 11 equals the expected expression for the J gene in cluster 14. The second column of this table shows you the p-values. They're all really tiny, like 0. Or 10 to the negative 7. Okay, so we might wonder, like if I'm a statistician and I barely know how to test this null hypothesis in a valid way, like what exactly is the software doing? The answer is the software is called Seurat. It's like very, very, very highly used in this community. The screenshot showing 4,000 citations is actually a couple years old, and I should update it. It has a lot more than 4,000 citations right now. This is kind of like the software. But if you sort of dig in to see how they're computing those p-values, they have the sentence that I think is really cute, where they say p-values should be interpreted cautiously as the genes used for clustering are the same genes tested for differential expression. So in other words, there's no kind of accounting for the fact that they've done double dipping here. And again, like in their defense, like the reason they're not accounting for it is because like statisticians haven't given them a solution. Like they know this is a problem. Like it's on us as statisticians to solve it, right? Okay. And just to be clear, I already showed you the screenshot of a review article saying that this is one of the grand challenges in single-cell data science. So this isn't just this one software package. This is just like the field has this problem. This is a real problem. Here's a quote from the review article, which I'll skip in the interest of time. So what exactly is the problem? Well, again, if I sample 100 cells, IID, cluster them, compute a p-value for the difference in means. Remember, my p-values are way too small. They should have been uniform 0, 1. They're not. So that table that is output with 20,000 p-values by this software, which shows me p-values like 10 to the negative 7, I am just not impressed, because I can get p-values like 10 to the negative 7 just like on normal 0, 1 data. So how can we solve this, and can we use data thinning to do that? And I also just want to say one thing, which just in the interest of time, I have not included in this talk in detail, so I'm going to say it verbally, which is sample splitting, though it's beautiful, does not solve this problem. You cannot get around this problem with sample splitting. And just in 2 sentences, the intuition behind that is that to cluster the data, you're operating on an object of length n, and then to test the hypothesis of interest, that also involves an object of length n, and it's the same object. If you split into an object of length n over 2, and you clustered and then tested, it would be 2 different objects. It just wouldn't be meaningful. So the point is, this is an example where you can't sample split. But as you're going to see, we can data thin to solve this problem. So here's what single-cell RNA sequencing data looks like. It's count-valued data, lots of rows, maybe 30,000 cells, 20,000 genes, positive counts, or rather non-negative counts. I'm going to go back to my table. I'm going to use Poisson data thinning, which tells me that for every element of this data matrix, I should generate a training data set by drawing from a binomial with size xij, probability epsilon, and the test data set is going to be x minus x train. Notice that the train and test data sets sum to give me the original data. My training and test set are provably independent, provided that I'm right about this Poisson assumption. And so that's the idea. I'm going to apply this idea to data from my collaborators having to do with cardiomyocytes. So they have some data where they're looking at cardiomyocytes, which are heart cells, and they're looking at how these heart cells differentiate. So you start out on day 0 with a whole bunch of heart cells that are just completely identical. And then over the course of 2 weeks, they differentiate into different types of heart cells. And so here's a figure from their original paper, where this is a representation of that data into 2-dimensional space, because the original data has 20,000 dimensions for 20,000 genes. This is a 2-dimensional representation. Each point here represents a cell in this 2-dimensional space. The axes here are labeled UMAP. UMAP is a dimension reduction method. We're going to just use PCA today because the added complication of UMAP is just not relevant to this discussion. But what you can see here, the different colors show me the day of differentiation. So like on the bottom left, we have all the gray cells. Those are the ones on day 0 before differentiation happens. And we can see there's kind of like a rainbow sort of from left to right as differentiation's occurring. So we can see that there's something really going on, like that first UMAP dimension is really capturing some type of actual thing that's happening to the expression of these genes over the course of the 2 weeks. Now, I'm a statistician. I don't use UMAP. I use PCA. So here's the same data, but if we instead apply PCA, and we can see, again, there's a rainbow. I've colored the rainbow now from red to green to blue to purple to pink. Well, I guess that's why I called it a rainbow. And so we can see that the first principal component really captures differentiation. So I believe that the first principal component of the data contains signal. The reason I believe it is because I can see with my eyeballs that there's a rainbow pattern, but again, I did PCA, of course, without using information about the day of differentiation. I just used the gene expression. And what I'm going to do now is I'm going to test the null hypothesis for each gene, that gene's expression is not correlated with the first principal component. So my null hypothesis is going to be that there's no association between the gene's expression and the first principal component. I do not think this null hypothesis holds. The reason I don't think it holds is because I can see the rainbow. I can see there actually is something interesting going on in the first PC. So we're going to just see what happens if we think that the null hypothesis doesn't hold. So on the y-axis are the naive p-values that I get if I don't account for the fact that I've double-dipped. In other words, that I'm testing the null hypothesis involving the first PC on the same data used to compute the first PC. So if you can read the y-axis, I know it's very small. The numbers go down to 10 to the negative 13. This is on the log scale. So these p-values are tiny. Each dot here represents a gene. So it's the p-value for a different null hypothesis for a particular gene. Those p-values are tiny. I am like rejecting the null hypothesis for like a lot of these genes. On the x-axis are the p-values I get with data thinning if I compute the first principal component using the training set and then test it on the test set. And actually what's interesting is if I can read the x-axis, it's also quite small, 10 to the negative 13 is the lowest point there. So the 45-degree line is shown in black, and what I see is that my data thinning p-values are not as small as the naive p-values, but they're still pretty small. Like as a statistician, 10 to the negative 13, 10 to the negative 11, I don't care. Like either way, I'm rejecting the null hypothesis. So the point is that my data thinning p-values, when the null hypothesis does not hold, are not quite as small as the naive p-values, but they're still small enough that I'm going to draw the same scientific conclusion. And in fact, if I want to understand why it is that my data thinning p-values are a bit larger than the naive p-values, what I can do is I can compute also naive p-values using only the test data. That's what's shown here in blue. And we can see in that case, the naive p-values and the data thinning p-values are essentially identical. So the only difference, the only reason that the naive p-values are smaller is just because it had access to twice as much data because it was able to use the training and the test set. So the point is that in a case where the null hypothesis does not hold, I can reject the null hypothesis with data thinning p-values. That's really good because a scientist wouldn't be interested if I said I have a way to give you a valid p-value and you'll never reject the null. They would like find a new statistician immediately. So, so this is just a sanity check that when there is signal, we can detect it. And now what we want to do is make sure that when there's no signal, we don't falsely detect it. So to do that, I'm going to look at only day 0 data. So these are cells only on day 0, and I've regressed out the covariates that are present in the data, which is just to say that, like, as far as we know biologically, all these cells are just like essentially equivalent. The difference between them is just noise. There's no signal here. And in fact, when we plot these cells onto the first 2 principal components, we just get a big blob. And now I'm going to show you a QQ plot. So if you can reach back into the recesses of your memory from like 40 minutes ago, I showed you a different QQ plot, and I said that the x-axis shows the quantiles of a uniform 0-1 distribution. The y-axis shows the quantiles of the actual p-values under the null hypothesis. And here I think that the null hypothesis holds because I think there's no signal when I only look at day 0 data. I would like to see beautiful p-values on the 45-degree line. And in fact, that's what I get with data thinning. And by contrast, the naive p-values are too small because they fall well below that 45-degree line. And so what this tells me is that when there's signal in the data, data thinning can detect it essentially as well as the naive approach. But data thinning is valid, whereas the naive approach isn't, because data thinning gives me uniformly distributed p-values under the null, and the naive method does not. And just finally, one last thing to say. In this single-cell RNA sequencing example, I used a model that said that each element of the data matrix followed a Poisson distribution. There's some disagreement in the field about whether that's a valid assumption or if you should instead assume some type of over-dispersed count model like a negative binomial. And I'm not going to wade into that scrum except to say that if you don't believe that Poisson distribution is valid here and you want to use a negative binomial distribution, go ahead, because we have a row in the table for that too. So just to conclude, everything that I've talked about today is a paper published on my website, or rather a published paper which you can find on my website, danielawitten.com, and then navigate to publications. We also have a lot of software tutorials. So when you're on this website, like linked next to each paper PDF, there's also a software page. And for many of these, there are tutorials that I really hope you find helpful. And if you don't, then send me an email and I'll ask my co-authors to help me improve it. So, all right, well, thank you so much. Oh, and one last thing, somehow the slide's not here, but like all of this work is joined with just like the absolute most talented PhD students. So if you look at these papers, like Anna Neufeld, she's actually faculty at Williams now. She just started there, but she graduated a couple years ago. Lucy Gao is faculty at UBC. Amir is a current third-year PhD student. Keshav Motwani is also a current third year. Yuchen Chen is starting a job at Hopkins as faculty. So these are all like the people who've worked in my group for the last 5 or so years. And so it's really special to be able to put all these papers on one slide because it really represents sort of the culmination of a lot of effort by a lot of really talented grad students. So thank you so much.